Hello to everybody. Welcome to Enjoy Science. It's Wednesday. It's 10 a.m. Central Time, so it's time to have fun on Enjoy Science. It's my pleasure to welcome today Dr. Florentia McAllister for a wonderful talk. And of course, it's my pleasure to say thank you again and again to all of you, because without you, the attendees, without your question and without your curiosity, such webinars make no sense. And also it's my pleasure to say hello to Liz Kent and to Laura Adame, who are all the time close to us, and also to a new colleague, Emily Lang, who will help us in Enjoy Science. And this is good news, because it means Enjoy Science is growing, expanding, and we have for you also the news that we will restart after this talk in the fall with the continuation of the series of Enjoy Science. Why? Because the speakers have done a wonderful job. All of them presented extraordinary, interesting talks, and all of you were all positive, and you attend in very large numbers this webinar. I have again to thanks to Giulio Draeta for organizing this, and also to Alessandra Ferraioli, with whom I co-host Enjoy Science. And with this, let's go at the center. So Florentia McAllister, our speaker today, it's a pleasure to introduce her. Why? Because she is a typical new wave of clinician scientists at MD Anderson, young, bright, extraordinarily well-trained in Johns Hopkins and also in Pittsburgh. And she is an associate professor in the Clinical Cancer Prevention Department and also GI Gastrointestinal Oncology Department. And she is focusing really on extraordinarily important topics, the topic of microbiome in very deadly cancers like pancreatic cancer. You know her very well. She's a wonderful colleague of us. You know her very well. If you are not here because she's publishing wonderfully well, a very high impact publication in this topic, and certainly we are all happy to have her here, and we are very curious about today's talk about microbiomes in tumors and beyond. So let's see what is behind this beyond. Thank you again, Florentia, and good luck with your talk. Please run the show.
Okay, well, thank you so much for the kind introduction. Let me get started with the sharing. Okay, so I think, okay, is it all good? Yes, perfect. Okay, so I'm going to talk today, I kind of made it as a sort of free talk with the title. I thought that I had to really enjoy the talk, so I didn't want to focus on one topic. That's why I call it Microbes in Cancer and Beyond. So I'm going to expand a bit on other related topics, which I find very exciting. So no financial disclosures. I'm going to start with my road to microbes. So as a physician scientist and oncologist trained in pancreatic cancer, it actually all started with realizing how hard it is to treat pancreatic cancer, and to see the very little progress that has happened in the past few, actually in the past decades, literally since surgery has started for pancreatic cancer. So I got very interested about the early tumorigenesis process. And since I trained in my postdoc prior to my clinical training in a host defense laboratory and studying basically the development of these type of T helper cells, Th17 cells, which are important for protection against different type of bacteria and also against fungal infections. I got very interested in studying these cells, because most of the risk factors for cancer actually are, especially pancreatic cancer, are linked to Th17 responses. Some of them are cigarette smoking, diabetes, obesity. So this really triggered my attention of trying to continue studying this type of cells, but in the context of cancer once I was in my fellowship. So these type of cells, we studied them in the context of different type of mouse models, and in a transgenic mouse model using pharmacological approaches or genetic approaches, we were able to see that if you block IL-17, basically you slow tumorigenesis. And this was independently also published by a second group in France using a different mouse model also blocking this pathway can delay tumorigenesis. We further along went to understand that some of the mechanisms related to this effect had to do with and sort of unexpected function of IL-17, because we think of IL-17 as a cytokine that basically recruit other immune cells, because it secretes chemokines through epithelium signaling, and it's basically known to recruit mostly myeloid cells, and that's how it fights infections. But this was sort of unexpected to see that in the pancreatic epithelium, specifically by sorting pre-malignant cells upon IL-17 inhibition, we saw that embryonic stem cell pathways were being altered. So this was very interesting to us, and we continue to have collaborations in this topic, in particular with BCLK1, which is also a target now for cancer stem cells and in cancer, in other type of cancer beyond pancreas. But the question that came after this is, what is the IL-17 driver? So why do we have IL-17 not only in pancreatic cancer but also in other cancers? It's associated with those risk factors that I mentioned, but it's not clear why. So one of the experiments that sort of solved that question was if we take stool specimens from patients with pancreatic cancer and healthy controls, and we put them on mice that have been treated with antibiotics, and then we look at the cytokines within the serum. IL-17 is at the top of the serum biomarkers on the mice that have received stools from pancreatic cancer patients, suggesting that the dysbiosis associated with pancreatic cancer development triggers an IL-17 response through changes in the gut. And that is actually easily to explain. This is a nice cartoon that I think puts it all together. So bacteria will translocate through the gut epithelium, will activate antigen-presenting cells like macrophages in this case, which in turn will secrete cytokines like IL-23, which is a known factor to differentiate T cells into a Th17 phenotype. And not only Th17 can produce IL-17, which are the T helpers or CD4, but also gamma delta T cells and innate lymphoid cells are also capable of secreting IL-17 and these other cytokines that are related. So these type of cells and these cytokines have already been linked to locally to the induction of tumors within the colon, which will make sense. They are secreted there, they are differentiated there, and they will have in turn an effect within pre-malignancy or within colon cancer. And Michael Karin and Cindy Sears have worked a lot on this. And this is just one of the publications that I just mentioned. This is work from Cindy Sears, and basically she shows in the APC mean mouse model that in particular ETBF, enterotoxigenic bacteroides fragilis, is capable of inducing tumors, which interestingly, it was a serendipitous finding she did while she was studying colitis and diarrhea in patients and in mice. And she had some mice that were left for a long time, and she actually discovered when she went to sacrifice it that not only they had more inflammation, but they also had more polyps, and that's when she went to study this topic. So not only she did that, but then she went to study the immune response in collaboration with Drew Pardoe's lab at Hopkins, and she actually found that IL-17 was mediating those responses. And I collaborated with that aspect because I was just joining my fellowship when this paper was happening, and it was fantastic how if you block IL-17 within that context, the protumorigenesis induced by ETBF completely shuts down, suggesting again in the context of colon cancer, there's a bacteria which triggers an immune response that is responsible for this tumor initiation. So I'm just putting this because I wanted to go from the gut to a very distant organ. So IL-17, if we think about these cells, not only will have an effect locally, but they will circulate. So they can affect distant organ, and this is to me the brain, the extreme of how you can have an effect systemically. So this is work that was just published by a group in Germany. And what they basically did is, I mean, we do know for a long time, work from Dan Liebman's lab has already shown that IL-17 is very important in driving basically immune responses and that are based on the gut microbiome. And if you have mice from different backgrounds, they're going to have different amounts of IL-17, with taconic mice having the higher levels of IL-17 because they have SFP, the main driver, and Jackson having the lowest. So we did know that for a long time. But this group went deeper into trying to understand if just replacing or just changes in the microbiota can actually rescue that. So as you can see here in the model of autoimmune encephalitis, the IL-17 knockout mice basically do not develop the disease. But then when they're co-housed with basically the wild-type mice, they actually rescue that clinical phenotype. Again, this is in a different type of model, but I think I mean, of course, this paper goes into a lot of details that I have no time to share today, but the fact that there's bacteria, which is driving IL-17, which is driving brain disease, very distant. So microbes are everywhere, I'm going to tell you now, and they're very variable. So I think the more I study microbes, the more I realize that the individual microbes are not really what matters, and what matters is really what's downstream of the microbes. What are the pathways that are being changed, and what are the mechanisms that might be common to several microbes? And that's why I wanted to share these few slides with you. So a group in Israel set up to understand the differences in species between patients with pancreatic cancer and healthy controls within the gut. And you can see here, right, you have differences. So they basically found that there's an increase in bacteroidetes and there's a decrease in firmicutes. But another group in China actually looked at the same thing and found similar changes. However, when you plot together the study from China and the study from Israel, what you can see is that the most clear difference is the geography difference. All the patients from China are at the top in the circles, and the patients from Israel are at the bottom in triangles. So in other words, geography, super important, and the variability in the microbiome between different studies done in different places is huge. That has also happened with the study done by our own Jean Wargo and the French group with Lorenzi Vogel and a group in Chicago, when they published that the diversity that correlates with immunotherapy responses is increased, but the specific species were very different between the three places. So all of this to say, microbial ecosystem coexist in our body. So we've been until now mostly talking about the gut microbiome and potentially systemic influences. But it's important to know that there are different types of ecosystems. The microbiome is not only restricted to the gut, and then even places which are thought to be sterile, that is in normal conditions. But in pathological conditions, and especially like in cancer, in which we know there are going to be a lot of pathological changes in barriers, permeability changes, vascular changes, hypoxic changes, then places that we are not expecting to have bacteria, they may actually get colonized. So I looked back at some point to understand if this is something new, and I found that this is not new at all. So William Russell, a Scottish pathologist that lived between 1852 and 1940, actually was looking at breast cancer biopsies. This was at the end of 1800, when he first evidenced what he described as microorganisms. Now, those may have been, well, the Russell bodies that went on to describe later, but it may have also been the first time that organisms have been described in cancer, and certainly triggered for several years further study in this topic. For some reason, after 1910, 1920s, this topic was sort of abandoned until the early 2000s, when the NIH Human Microbiome Project started, and a lot of interest in the microbiome sort of research. So talking about unexpected places like carotid plaques can host microbes, bladder cancer, and recently it has been shown that not only lung cancer can contain bacteria, but also these bacteria are very important in driving this tumorigenesis. And this was work presented by Jean Chen. She was giving one of the talks at MD Anderson in last year's symposium. And she actually did really a lot of functional work when she was at Tyler Jack's laboratory. So this is one of the tumors in which we do know that the bacteria present on it has a functional relevance, and it's mostly mediated and affected by gamma delta T cells. So getting out from those few tumors unexpected, and the group from Israel, mostly led by David Strussman, looked across tumors, and they basically reported that most tumors contain bacteria. And you can see here that tumors like GBM are mentioned, and breast cancer. So certainly pancreatic cancer was of the most interest to us. And this is just some of the staining that were done by the same group in another publication. So they did fish staining, as you can see in the middle, to detect 16S, and then also LPS staining, and they certainly saw correlation between these two sites. Next is, so far we mentioned the links between microbes and IL-17 and the fact that there are microbes that are variable and that basically can be anywhere in pathological conditions. What I want to tell you now and try to convince you is that microbes are not so only present in those places, but actually they matter. So in terms of preclinical work, this is the first paper I will say with pancreatic cancer in preclinical models that show that microbes are important. This is work from the laboratory of George Miller. They basically took the KPC mice and they re-derived them in germ-free conditions, and they found that there's actually a very strong delay in tumorigenesis. When those mice receive feces from a wild-type mouse, you can see how some of the ADMs are sort of increasing, so there's a bit of risky of the phenotype because they're putting some microbes, but when they actually do transplant with feces from a P-type mouse, there's a whole complete rescue of the phenotype. And this basically shows that microbes are certainly required for tumorigenesis. A second group around the same time, led by Chris Jobin, showed in a different mouse model also the absence of microbiota delays tumorigenesis. And in a totally different model, in a subcutaneous implantable model, the group from Ashok Saluja have also shown in the same year that using antibiotics, different approach, can also delay tumor growth, and then this effect is actually lost in the RAG1 knockout mice, meaning that you do need the activated T cells for this effect of the antibiotics to happen. So you have the microbes, and you have an induction in tumorigenesis, and this effect is T cell mediated. This is actually functional work that was produced again from the same laboratory that showed the presence of bacteria in the pancreas from Ravid Strassman in the Weizmann Institute. And again, I feel like now by giving this talk, I'm realizing most of these findings were serendipitous. The way he ended up doing this work is he was doing screening, drug screening, and he realized that if there were some cells that consistently were having a very unexpected response to gemcitabine, ended up being that those cells had mycoplasma contamination. So then he expanded that work from the mycoplasma, instead of throwing the cells away, to other bacteria that actually were important, like, for example, gamma proteobacteria, which is present on pancreatic tumors. And he realized that indeed the same mechanism was also happening with other bacteria. So here, this is a very simple experiment in which he injected these colorectal cancer cell lines in mice in two different flanks, and basically added bacteria to one and did not add bacteria to the other. And you can see how when he treated with gemcitabine, basically the presence of bacteria does generate resistance. And when you have no bacteria, then the drug can actually be effective and shut down the tumor growth. He went to try to understand what was the mechanism for this. And for this, he actually focused on one enzyme, which is the cytidine deaminase, because of course makes sense. It is the enzyme that basically metabolizes gemcitabine. So he showed using an in vitro system that only treating with gemcitabine or only treating with ampicillin did not do much in terms of cell killing, in this case measuring apoptosis, which is type A staining in brown. But if he combined gemcitabine with ampicillin or gemcitabine with chloramphenicol, the synergistic effect was very obvious. And then to prove the specificity of the enzyme, he went to delete the cytidine deaminase from these bacteria, as you can see here, and you can see that if you give gemcitabine on the tumors that have had the bacteria with the enzyme deleted, then the gemcitabine basically is now effective, meaning that that enzyme was inducing resistance to this chemotherapy. So this is very interesting, and I'll come back to that a little bit later. This is a paper very, very important from Susan Bowman, which she developed when she was at Harvard. She's just recently started her own lab at the Fred Hutch. She basically looked at microbes within colorectal cancer and liver metastases, and he tried to find correlations between them. And what she observed, and you can see that when she made this kind of nice design comparing them, but also here in the taxonomic analysis, is that the patients that did have, either by PCR or by culture, fusobacteria within the primary tumors, they also had it within the liver metastasis. You can see patients sort of 1 to 7, and then patients 10 and 11, they did not have it on the primary, they did not have it on the metastasis. And you can see here, it's basically the red color is the fusobacteria. So if it is on the primary, it is on the liver met. except this exception. But in all of these other three, also if it's not in the primary, it's not in the metastasis. So this paper strongly suggests that the bacteria or the microbes might be intracellular, which of course is accompanied by other data that they have done with electron microscopy, that the microbes can live inside the tumor cells. And then upon metastasis, those cancer cells can also carry along the bacteria, which is very interesting. There are other alternatives to that, which could be that the bacteria could be coming through circulating factors like exosomes and also as free DNA. But this was very interesting, very important in terms of the relevance of microbes in metastasis. So we asked the question if the tumor microbiome could be different. In our case, we don't have responders and non-responders in pancreatic cancer, but we do have patients who go to live very long time. We call it long-term survivors, and they are very unique patients, but we look for that cohort here, and we had a second cohort, Johns Hopkins. And interestingly, the group from Johns Hopkins with Laura Wood, Anirban at that time, and Bert Vogelstein looked at those two cohorts in terms of genomics, and they actually found no differences from that perspective. So which was kind of more encouraging for us, that if it's not the genes, it might be something microenvironmental. But we thought if there's no difference in the tumor microbes between these two groups, it's very likely this does not have a translational relevance. So we did 16S sequencing in these two cohorts. And what we found was initially in both cohorts through different type of indexes, we found an increase in the diversity, microbial alpha diversity, which is really a measurement of microbes heterogeneity, the number of species. So we have seen in the past other papers reporting that higher gut diversity was correlated or was predictive of benefit in different tumor treatments. But in this case, we are seeing that within the tumor, also the diversity is important. The second thing we did is we looked at beta diversity just to compare within samples from all these patients. And you can see clearly the differential clustering in both the MD Anderson and the Hopkins group. But I thought even more interesting is when we put them all together, all the patients from the two groups, from the two places, we found that the long-term survivors sort of clustered together, suggesting some potential microbes that are enriched in these patients, and those are the Pseudoxanthomonas, Saccharopolyspora, and Streptomyces. Again, we found these, but again, because I see all along all these differences in geography, I think certainly it is important that these, Hopkins and Houston are distant, and certainly these were the bacteria that were found in these two groups. I'm sure that if we continue studying in other places, we may actually find that may not necessarily be the same bacteria. Though I should say, through other studies, we find that the tumor microbiome is a lot more stable and less subject to environmental changes than the gut microbiome. So again, we have to prospectively validate this, and it might be that these are the relevant ones. This brought us to the next question, which is, okay, if the microbes matter within the tumor, how can we change them? How can we modulate that? So to answer that question, we actually did a very short study with the help of the surgeons here. In a very short time, we're able to collect tissue from the tumors, adjacent tissue, and also stool from the same patient. And certainly all of this was also supported by the Moonshot and the Pancreas Center, so was done in a few months. We did 16S sequencing, and by looking at the origin of the microbes, we were able to answer this question. As you can see here, if you focus on the tumor, you can see that 20 or 25%, which is the green color of the microbes, are actually coming from the gut because they're present on the stools. Around 25% are also present in the adjacent area, which means that perhaps were there before or perhaps are related to inflammation, etc. The 50% that is in gray, it's not on the normal adjacent. I shouldn't call it normal. It should be like adjacent tissue and gut. There might likely be coming from the upper GI, and now there's been a couple of papers in the last few years that actually mention that the oral microbiome and also the duodenal and stomach microbiome may actually be very similar to the content of the feeder microbiome. So this fundamentally told us that we could actually change the tumor microbiome by changing the gut microbiome. So if we change that stool, at least 20-25% of the composition of the tumor can change. And if we do it in time, we might be able to increase that even further. So we built this platform we called microbial humanized mouse. And we actually took samples from 3 types of patients, from short-term survivors, long-term survivors, and healthy controls. And in this case, the big difference is with the long-term survivors that I mentioned previously in the studies. These patients were, some of them in the study, but of course, they had cancer in the past. Now they have no evidence of disease. That's why we know they're long-term survivors. So we thought that in some ways, the long-term survivors and the healthy controls are pretty similar because they're basically patients with no cancer. The only difference is that the LTS had cancer remotely, like more than 5 to 10 years ago. So with that model, we set up to answer the question, can we use FMT to modulate the gut and tumor microbiome? And we use the same mouse that I described before with the 14 days of antibiotics, then the transfer of the fecal matter, and then 2 weeks after, we did the implantation of KPC cells in the pancreas, and then we waited until day 35 to finish the experiment. The first thing we did is we looked at the colonization, and actually even, we found that sort of shockingly that the difference between the 3 groups was huge. Again, because of the variability in that microbiome, I was not expecting that with a few number of donors. We are using in each of the studies 3 donors that go into 3 to 5 mice, but you can see the clear cluster differences between the 3 groups of mice with the 3 different types of donors. We then went to look at the tumor microbiome, and again, we were able to see very differential microbes within the tumor between the mice that received the stool from the different type of donors. So with this in mind, we're able to stably colonize the gut and the tumor. We went into our next question, which is, can we now alter the tumor immune activation status? And for that, we did several types of immune profiling, and just here, simply showing a few of the cell types. You can see that the long-term survivors, when they went into mice, the stools, they triggered basically an increase in the immunoactivating cells, like CD8 T cells with interferon gamma secretion, and they actually decreased the number of T regulatory cells and MTSCs. And the opposite happened with the mice that received stools from patients with pancreatic cancer. They had the lowest number of cytotoxic T cells, and they had the highest number of immunosuppressive cells. So with this in mind, the idea that the microbes can actually transfer the immune activation status of the donor, we set up to ask the last question for this story at least, which is, can then the bacterial modulation affect the tumor growth? And indeed, we found that it did. Not only the PDAC stools resulted in largest tumors, but also the long-term survivors resulted in smaller tumors, which we were not expecting. So basically, this sort of, at least we can hypothesize that there might be some bacteria that is present on patients with pancreatic cancer as part of the dysbiosis that may contribute with that process of basically more like negative bacteria. On the other hand, the long-term survivors may actually have some beneficial bacteria. And then we finally blocked CD8 T cells, and we actually found that we actually lose some of the effect of the long-term survivors, meaning that, again, T cells and CD8 T cells in particular might be mediating this effect. So these sort of links with the immunoprofiling that we found on the donors. I didn't show this before, but basically, as other groups have reported, the amount of T cells, it's actually correlated with survival. So we clearly found that the survivors have higher number of activated T cells, lowest number of Tregs, MDSCs, and macrophages and others. But then when we did immunoplex to also show some type of spatial differences, which I'm not going to show now for because of time. But there's a publication also from Steve Leach, who showed a few years ago that the patients with, basically, they also looked at long-term survivors in the Memorial Sloan Kettering hospital, and they did not focus on microbiome, they focused on TCR sequencing of these patients, and what they found was an increase in the neoantigen quality that correlated with survival. And very importantly, those neoantigens were mostly of microbial origin. So this suggests potential cross-reactivity as one of the mechanisms for this effect. But that, of course, has to be validated. So going back to IL-17. So is bacteria-driven IL-17? Now I sort of linked, started with IL-17, ended up with bacteria. And the question is blocking, if we block IL-17, can we actually find a similar effect to altering bacteria? So back to what I showed before, this is sort of the slide linking my story and how the PDAC FMP increases IL-17. So we thought if we block IL-17, we should be able to modulate pancreatic tumor growth in an orthotopic model. However, that did not result in changes in tumor growth. When we put the tumors, the KPC, and we basically established tumors, and then we treated with neutralizing antibodies against IL-17. But what we did is at 2 weeks, we did RNA sequencing and immunoprofiling through CyTOF and other methodologies. And through them, what we found was that the expected cells, usually driven by IL-17, were being modulated in this model. So we did have lower number of myeloid cells and neutrophils in the mice that received monoclonal antibodies against IL-17. What we went to look after this was what happened with other cell types. And what we found was that the CD8 T cells were increased in the mice that had been IL-17 neutralized, but further along, those cells were actually closer to the tumor cells. So we thought that potentially this could be a mechanism that may maintain the T cell exclusion, which is so well known in pancreatic cancer. So that was sort of encouraging. We further along looked at levels of different types of checkpoint molecules, and we found a correlation with PD-1, which brought us to the next question, which is, okay, if IL-17 inhibition itself cannot phenocopy the bacteria modulation, can it actually reverse immunotherapy resistance if we combine with other checkpoint blockade molecules? And that's what we did. We combined IL-17 with PD-1, and you can see here in a subcutaneous model, the effect is impressive, the synergistic effect between blocking PD-1 and blocking IL-17. In an orthotopic model, we were also able to increase survival on these mice. And then finally, we also did a genetic approach to this by deleting the IL-17 receptor from the KPC cells, implanting them in the pancreas, and you can see how actually on their own they do have some decrease in the tumor growth, but then in the combination with anti-PD-1, basically the tumors were completely rejected. So we actually thought after this, if IL-17 drives the neutrophils and ultimately there's a sensitivity to immune checkpoint blockade, why is this happening? Like, what are the neutrophils doing to this? What is the mechanism for that? So we got interested about NETosis pathway because this is one of the, it's a cell death typical from neutrophils in which they basically release the granular contents and the chromatin in the extracellular space, and they form these sort of nets as neutrophil extracellular traps. That's why they look like traps. And they're associated with innate immune responses to infection because, again, the whole point is the neutrophil dies, and by dying and releasing these traps, it's also sort of physically stopping some of the microbes, which in turn might be killed by some of the released granules from other live cells. So what we did is we set up a similar experiment, but initially we wanted to know if there could be an effect of IL-17 on NETs. And for that, we initially added IL-17, TNF, and IL-6, which are other, TNF is a known NETosis inducer. And what we found in gray, as you can see, is that IL-17 does not change NETosis. In this case, we are measuring NETosis by net extension on the neutrophils. It does not change NETosis induction. However, if you first expose the KPC cells, the tumor cells, to IL-17, you collect the conditioned media and you add it into the neutrophils, then you see an induction in the NETosis. And we are still trying to figure out what is in the conditioned media that is driving this process. But we then went to patient samples, and we basically exposed human neutrophils to serum from patients with pancreatic cancer versus healthy controls. And what we found was an increase in the NETosis capacities using serum from patients with pancreatic cancer. Here we are using PMA as a control, which is a known inducer of NETs. So with this in mind, we went back to the mouse model, and initially we neutralized LY6C, which is a marker of myeloid cells, and it can be used for neutrophils, also neutralization. And you can see how actually on its own, it decreased tumor growth, and there was not much difference when it was PD-1 was added to that. And then when we use the PAD1 for knockout mice, PAD1 is actually, PAD4 is the enzyme that is required by the neutrophils to make NETs. If the mice do not have this enzyme, so they cannot make NETs, they do have some, again, there's in this case mild effect, almost no effect, but with addition of anti-PD-1 on these mice, there's a very synergistic effect, sort of implying that at least one of the mechanisms for this IL-17 might be the effect, might be the recruitment of neutrophils and the induction of these mitosis. So the question is now, can we modulate the gut tumor microbial axis for therapeutic purposes? Everything that I've told you so far, it's about mostly mechanisms and connections between IL-17 and microbes, but can this actually help patients? So this is sort of my mental working model. So we have protomorgenic bacteria, or sort of negative bacteria, and then we have anti-tumorigenic bacteria, or potentially beneficial bacteria. And again, when I'm saying this, I don't mean there's one species. I mean there might be communities that may do this. So at the end of the day, it's the balance between those two types of bacteria that will end up sort of moving this scale into a heavier immunosuppression or heavier immunoactivation. So the goal should be to move that into an immunoactivation, of course. And I see that there's really like three approaches for this. You have the antibiotics, which you want them mostly to decrease protomorgenic bacteria. However, we know that's going to be hard because most of antibiotics don't have a very narrow spectrum. And if they do, they may not alter all the protomorgenic bacteria. The second approach is the use of single bacteria or consortia. So you could think, for example, in our work, if we could use the same bacteria that we found in the long-term survivors, and we actually do experiments with them and see if they have a direct effect. So far, most of those experiments that we have done, we have not found that there is a direct effect of those tumor bacteria. And the third approach will be the fecal microbial transplants. The goal will be to do both things, right? We want to, and especially because FMT is always preceded by antibiotics. So you could use an antibiotic that mostly decreases your protomorgenic bacteria, and then with the FMT afterwards, you replace mostly with beneficial bacteria. So just a few slides about this. So about the antibiotics, when we started doing this, we thought we should look, at least in a cohort of patients treated at MD Anderson, if historically there has been any benefit of taking antibiotics. And we looked in more than 700 patients, metastatic and with resectable disease. And what we found was that the metastatic patients, actually the resectable patients, the patients who actually went for surgery, did not have any change in survival based on the antibiotics intake, which sort of makes sense because when a patient goes for surgery, it's the surgical outcomes that usually are more important, like the margins or the lymph nodes, etc. But in the patients with metastatic disease, we did find that the patients who took antibiotics for more than 3 days actually had better outcomes. And very important to us, when we look what chemotherapy did they take as first line of chemo, we found that gemcitabine-based chemotherapy was associated with the better outcomes, which actually makes sense based on the preclinical data that I showed before from Ravid, in which he showed that the bacteria are mostly interfering with gemcitabine metabolism. So I think this was sort of a nice clinical validation of that preclinical study. And there are now ongoing, especially in Pittsburgh and in New York, there are 2 clinical trials that they're basically using gemcitabine in combinations with antibiotics. In fecal microbial transplant, we just have these 2 papers that came out, one from the Israel group and the second one from Pittsburgh. In melanoma patients that were refractory to immunotherapy, FMT actually was able to overcome that resistance in 30 or 40% of the patients, as you can see here, with the 3 of the 10 patients sort of responding. And they looked at the immune responses, and they seem to have at least a very good correlation between the patients who responded and the immune activation status. I think importantly here, the red color lines were donor 1 and the blue is the donor 2. And if you see the responders all received the FMT from the same donor, which again, it's a low number. It certainly doesn't have enough statistical power, but it sort of suggests that it might be a relationship between the characteristics of the microbes found on that donor. And again, these were 2 pilot studies, but I think there's going to be a lot of work done on these fields in the terms of randomized trials to sort of understand if this is a real signal. There are 3 FMT studies that are being done here at MD Anderson. One is the one we are doing, which is basically with Anivan Mitra, with Jen, work with Mimi, Dr. Bhutani, and others. We're mostly looking, and certainly Dr. Katz, who is the surgeon for the study. So we're basically doing FMT prior to surgery with the main question if we can change the tumor microbiome and can we change immunosignals. So it's really a sort of phase 0, very small study about to start now. The second one is being done by Ginny Wang, and she's mostly looking at the use of FMT to transfer mostly healthy controls, stools, into patients with colitis from immunotherapy treatments. And then the third type of trial is led by Dr. Overman, and he's using FMT for colorectal cancer, similar to the melanoma study I just showed. So in the last few seconds, which I don't know how much I have, I think I have more than seconds. I have maybe like 5 minutes, so there's enough time for questions. This is something that sort of got started last year, so that's why I called it and beyond, because this is sort of where we're expanding now. So as I mentioned, the IL-17 connection with NETosis, when the COVID-19 started, Michaela Eglevat, who is the senior author of this paper, started tweeting about NETs in COVID-19, mostly present in inflammatory sites, but also very important in vascular sites and sort of responsible for some of the microthrombotic events, very common, especially on young patients. And since I'm not in Twitter, but I have a good friend who is on it, with Anivan Maitra, he connected me with her, and we actually started this network with all of these people who were all over the world, and they were all studying NETosis, and we sort of met bi-weekly and started sort of having discussions about how we target NETosis from very different approaches. So from our perspective, IL-17 and Th17 responses have been described to be associated with COVID-19 responses, especially in the stage 3, in the more severe critical patients, kind of at the time of the cytokine storm, and sort of also leading into the stage 4. One of the things we were able to do through this collaboration is Rob Schwartz, who was working in the ICU in New York, and actually getting lung organoids from COVID-19 patients, he did single-cell sequencing, and we basically asked him, can you look at these IL-17 target genes within the organoids? And he did look at them and actually found that most of the target genes were upregulated on these patients, which was kind of encouraging for us. The next thing we did is, with help of our collaborators here and also with Syed Payman, Mogadam, and also with Biri Chandra, who is a grad student in the lab who actually got excited about doing this. She also participated in the PDAC work, but she thought it would be fun to look at this. She used lung epithelial human cells and basically added these different factors, mostly IL-17 in this case. And similar to what we found before, the IL-17 alone will not do anything to the NETs, but if you add IL-17 on the human epithelial cells and then you add into the neutrophils, then you see an induction in NETosis, similar to what we found in the pancreatic epithelial. So the idea will be, if you block IL-17, you have kind of a dual effect, which is you're blocking the pro-inflammatory effect of the cytokine storm that is characterized by these neutrophils being recruited to the lung, but also you can block NETosis not only at the lung, but also systemically, which could also result in decreasing the amount of thrombosis. So based on that, we started writing this trial with Pinar in my lab, Canligi Claire, who is worked with me very closely, and she's the clinical research scientist who actually wrote this trial really quickly, and Methodist has been working on it, and I think we're hoping to finally, after a long time, get it started, but it finally got FDA approval. So my conclusions for today are: number one, I think I could say tumor microbial diversity, the signature of bacteria, and immune activation status, more importantly, are predictive of survival. In other words, tumors, microbes really matter. Second, there's a GAD to PDAC microbial axis which can modulate the tumor immune activation status and also the tumor growth. Th17 cells differentiation, which is triggered by the GAD or tumor bacteria, can induce and maintain tumor suppression, and at least partially by neutrophils and NETs. And I think adding to these Th17s are not only important, even though they are generated within the GAD, they are very important systemically. And finally, IL-17 neutralization may play a role in COVID-19 treatment. We will see through the secukinumab trial. And actually, I have one more, which is the most important, actually. Microbial modulation may represent a novel strategy for cancer therapy, very likely in combination with other modalities. And I think with this, I'm going to thank everyone. This is my lab celebrating Christmas. So everyone here who have contributed in one way or another to what I presented today, my collaborators at MD Anderson, especially Annie Van Meiteren and John Wargo, and my collaborators outside MD Anderson, and of course all the funding sources. So I'm here for questions, and I think this is it. I'm going to stop sharing.
Thank you, Florencia. It was a wonderful, wonderful talk. Really very, very interesting research. And in fact, let's start with the question. So the first one is a more technical. How do you control for specimen contamination?
So, yeah, that's a great question. So in particular for the tumor microbiome study, so there's two types of ways we do that. When we looked at the tumors, what we do is for each of the tumors, before starting to analyze even, we looked at the non-tumoral area. So for example, if you're looking at paraffin, we will have to, each patient provide its own paraffin. So we have like paraffin control from the same sample, and we also have paraffin control from non-related samples. So we will build like a filter made of the contaminants, basically, and then we can actually filter that from whatever we are actually detecting. There's also other known contaminants which we will have already preset from the literature. So we have actually, between the ones we detected and the ones which are known contaminants, we made this filter and we run it after doing the sequencing. That's one way. That's how we do it sort of for the 16S computational analysis. But the other thing is, for example, for the study on the patients, I think the best proof of specificity of our findings is the fact that when we did the tissue adjacent to the tumors, we were not able to find the microbes which we found in the gut. I think to me that is a very important proof, because if there will be contamination, there should be cross-contamination in both at the tumor and at the adjacent. So I thought that was a kind of good control within the sample design.
Thank you very much. Another question. So besides the regulation of the tumor immune microenvironment, have you evaluated if soluble factors actively secreted by bacteria, metabolites, bacterial components, molecules, can directly affect tumor cell biology and also tumor microenvironment cells?
That's a great question. So we are looking at that. One of the things we've done, and actually was included on our publication, was we predicted metabolic pathways that were altered based on the microbes, using like microbes. So that's just a prediction. And we did find that, pathways that have to do with the amino acids, that have to do with xenobiotics, were altered by the LTS bacteria. But what we are doing now is we're actually measuring the systemic. We are doing metabolomic studies and looking at that. So we do have some of that data. It's yet too early to mention what are the pathways, but there are metabolites indeed that we are finding correlating with the long-term survivors, and we are hoping to study that a lot deeper.
Thank you very much. Now a more complex question of three parts. So, first part, do tumors modify gut systemic microbiome? For example, generators sustain selective advantage for protumorigenic microbiome population. So tumors modify gut systemic microbiome.
Yes.
And then related to this, do non-tumor tissue microenvironment also contain microbes, and this influence tumorigenesis?
Yes. So I'll start with those two. So yes, I do think that the answer to the first question is yes. I do think that the tumors can modify the gut and systemic microbes. So in other words, I think, again, that was shown. There's at least 3 papers showing after KRAS activation, there are changes in the gut. So we do know that from the preclinical studies. And then in turn, that gut microbiome can also affect the tumor microbiome. So it is going in both directions. And then to the answer to the second question, if the non-tumor tissue microenvironment also contains microbes, we found this within our model. We have not done, of course, because you don't usually get tissue from like very distant places, right? But in mice, we have looked, and there certainly seem to be some of the bacteria that we found within the primary tumors that also they have that correlation with the gut. But we have not done the experiment of looking, like adjacent and then far away. That will be the experiment needed to answer that question, which we have not done. But I'm expecting that because it's that orange portion of the curve that I show of the graph, that if there are some commonalities between the tumor and the adjacent, those might be playing perhaps a role more in like the tumorigenesis process.
Thank you. And related to these two questions, it's this one. Is there any selective tropism for pathogenic microbiome to tumors?
Yes. So I think we are looking at that. I don't think this is a super interesting topic. We have some hints, but we have nothing final. Otherwise, I would have definitely talked. But I do think that there is tropism. And I think, especially if you're thinking that the bacteria is intracellular, there might be a contribution of the, depending on the type of cell, on the type of microbes, that allows some cells to sit in one organ versus another. Also, if we think that the microbes are not going in the cell, but they're actually going with immune cells or exosomes, etc., they may actually be part of the pre-metastatic niche that might be giving rise to different type of mets in different organs.
Thank you. Now, another question. So, great talk. You mentioned that the geographical differences will impact microbiome population in tumors. But would it be possible to extrapolate a gene signature of the bacteria present in the tumor that would be predictive of the immune state of the tumor?
Yeah. Well, again, if you look at the papers, each paper has its own correlation between their species and the immune pathway. What I could say is, I think it would be important to look, for instance, what are the common pathways between all these studies? What metabolic pathways are in common, for instance, and how, and think about targeting ultimately those pathways rather than, changing those bacteria. Yeah.
Great. Another wonderful, fantastic talk. Can you please comment on the effective and unbiased approaches to detect and profile intratumoral bacteria and their interaction with tumor and the host immune system?
Yes. Well, I mean, there are several. So if the question is about methodology, 16S sequencing, of course, we know there are a lot of limitations. At the same time, for some applications, I mean, if you're talking about places with a high biomass, like if you're talking about gut microbiome, certainly other approaches like next-gen sequencing and just getting the basic or the whole exome, or even so any technique that will allow you for whole sequencing of every gene that you have will be more important than 16S, right? But I think when you are dealing with organs in which there's such a low biomass, if you're applying those techniques of sequencing everything like next-gen sequencing, you will end up with 99.5% of human sequences. And then with the 0.5 that you have, yeah, you can look at things, you might be able to compare some, but you cannot really do a very good profile. Plus, imagine the cost of having to, you're basically throwing away 99% of the sequences. However, this is very important to do retrospectively and this is something that we're doing now with Nadim Ajami and with Jen and people at the Prime TR, is, reaching out to scientists who have already done that. And, since you've already done the sequencing, we can now look at that, percent of microbes within the sequences. But prospectively, I would say that's very difficult. I think there are going to be more techniques that have to do with selecting. So there's going to be like a capture sequencing in which you're mostly getting rid of the human and you're amplifying, mostly the microbial sequences.
Thank you. Again, another question. Thank you for the great talk. You hit very well, Florencio, on this geographic distribution. This is also about this. So, regarding this geographic distribution, can you speak about the downstream mechanisms that are probably involved in this stuff?
Yeah, I think that's very similar to the previous question on the geographic implications downstream. As I said, I think metabolic pathways might be very important. As I said, I think we should, first of all, this points to the fact that we can't just do these studies at one place. They have to be multi-institutional, and we have to sort of work together and sort of to get away from which species did you find. And because, it's a lot more exciting for a scientist to get to find one bacteria and to do all the mechanistic, studies and sort of but you're really, narrow tunneling yourself into, that one path, which might be real, but it might not be important, relevant, and, applicable to other places. So I think by looking across places, across institutions, across countries, and mostly define what's downstream of those communities that you're finding. And I think now that most of this data is it's public access, we should be able to do this more and more.
Thank you. Another question. What are the challenges of adding analysis of tumor bacteria to clinical trials? Is an interest in doing this?
Yeah, well, I think a lot of places are doing this now. So certainly, there's going to be, sort of the variability. It is going to be, of course, an issue. But I think places, like certainly at MD Anderson, I know for most of the platforms, this is being incorporated now. So it's not different than most of the storage that is used for other applications. It's basically like you want a fresh froze as soon as you can. That's kind of the simple answer.
Thank you. And let's take one more question. Are the bacteria in every cancer cell, or are they only in some of the tumor cells?
Yeah. So again, I think there's only been a couple of papers on this, and they're very specific. And most of what they've done is mostly in vitro work, and it's using electron microscopy. So, it has been, in some of the studies, it's been shown that it's the cancer cell that can contain bacteria. And in others, it has been shown that mostly like macrophages can contain bacteria, which of course we knew, but I'm talking within the tumors. I think more important than that is which one actually matters, right? Because if you find bacteria in the tumor cell, the question is that the one that is actually driving the process, or it's the one that is on the macrophages, which we knew all along, because macrophages, will capture bacteria everywhere. It may not necessarily have to do with the tumor microbiome itself. They might be even generated in other places and travel through the tumor.
Thank you. And now my time for one question. So Florencia, the people are wrongly considering that non-coding RNAs were discovered in 90s and 2000s. The truth is they were discovered in 80s in bacteria. Now there are studies showing that non-coding RNA from bacteria influence the human tumors. So the contrary concept of non-coding RNA field.
Very interesting. I think this is something that we could keep chatting between you and I, and we may think about potential ways to assess that. I honestly never went as far as thinking about that, but I think it's a very interesting concept, and I do think that people have been looking at this not in the context, of course, of tumor microbes, but yes, in the context of immune modulation, which ultimately might be modulated by these microbes. So yeah.
Thank you. So with this, I want to thank you very. It was a wonderful talk, excellent data, and very, very interesting data. I know a lot of questions. Now I want to thank also to all the attendees to tell you that for the summer it's a break, so let's enjoy summer, let's enjoy life, and of course let's make discoveries. And we will come back on Thursday, September 9th, and we will be in touch with all of you. And also one point that come out now. Thank you, Florencia, for wearing the Enjoy shirt, Enjoy t-shirt, Enjoy Science t-shirt. It was a hit. I got a lot of emails, SMS here in the chat. So, of course, all of you are very welcome to request such shirts, and I will speak with Liz Kent to giving you some information. And basically, Liz was very kind in offering these shirts to all the speakers, because this is the minimum what we can say, to say thank you very much to all of you, and to say that in fact Enjoy Science, it's what it's our hobby and what we love more. With this, thank you, Florencia, again. Thank you, all of you, and looking forward to see you in September. Keep making discoveries and be happy. Thank you again.
Thank you so much again.